This video will help you to learn proper aseptic techniques while working with the bacteria. It will also help you to learn techniques for sampling, inoculating, and staining bacterial samples. It's very important when working with the bacteria to follow the lab rules and use aseptic techniques to prevent any contamination from cultured bacteria. You will sample the bacteria from 2 different sources and will grow them on 2 different types of media, the TSA media and NA media. Take a test tube with 2 sterile swabs and go outside the BLC to sample an environmental source. To inoculate, open the lids of Petri plates few inches and begin to swab, going from the side and swab back and forth across the surface of the media. Turn the plate 90 degrees and do the same thing. Close the lid and discard your swabbing graduated cylinder. Repeat the same process for the other type of the media. Now you have a sample of environmental bacteria on 2 different types of media. Be certain that you inoculate the media of the Petri plate and not the lid. Next, collect a sample of bacteria from your body using sterile cotton swabs and 2 Petri plates. The best place to collect the body samples are behind your ears, under your fingernails, and on your hair. Inoculate your plate using the same process that you use for the environmental samples. Discard your swabs in a graduated cylinder. Be sure you label your plates, environment, or body and turn them upside down. Place the plates in the 2 different temperature incubators as indicated in your lab manual. After your cultures have grown in the incubators, it's time to sample and stain the bacteria and view them under a microscope. First, disinfect your area. And make sure to let it air dry. Then get a microscope slide and one of your bacterial cultures. Light your Bunsen burner and using an inoculating loop, put a loop full of water on your slide. Run water down the wire and the loop will hold the right amount of water you need. Next, flame the loop to the redness by putting it at the top of central blue flame. Cool for 5 seconds. Open the Petri plate and use your loop to scrape the surface of a bacterial colony. Don't take any media or large bits of bacteria. You will have plenty even if you can't see it on the loop. Place the loop into the drop on your slide and spread it around the slide surface. Let the slide with the bacterial sample air dry. Reflame your loop and repeat this procedure for the other 3 bacterial colonies. Be sure to label your slide to correspond to the source of bacteria, type of media, and incubation temperature. After smears have air dried, heat fix them by passing the slide 2 or 3 times over the flame. Hold the edge of a slide and tilt this slide up so the flame doesn't wick back and burn your fingers. After making smears of your cultures, you will stain the bacteria by using gram staining technique. Follow the step described in your lab manual precisely. You will be using 3 different stains and several washes to do the gram staining. Timing is really important in gram staining, so you may want to work with a partner to help you to keep track of time. Work with all 4 of your slides in a staining tray at once. First add crystal violet and leave for 30 seconds. Wash with distilled water. Next, add grams iodine and leave for 30 seconds. Wash with the colorizer for a few seconds until most of the purple washes out. Immediately wash with distilled water. Add the contrasting saffron in and leave for 30 seconds. Wash with distilled water and gently blot the slide dry. The best way to observe bacteria is through compound microscope by using oil immersion lens. Use your gram stannous light and observe them at progressively higher magnification using compound microscope. Be certain that you can clearly see well-focused bacteria under the 40x objective before proceeding to oil immersion. Once you have added oil, you cannot go back to lower magnification, or the oil will remove the stained bacteria from your slide. Follow the procedure in your lab manual for recording the microscopic and macroscopic appearance of your bacteria. Disinfect your work area and turn in your cultures 4 slides and 4 plates as instructed.